Um, Welcome everyone. Uh, Today we'll be hopefully exploring how synthetic biology, a field that fuses engineering principles with biology, is enabling us to design new biological systems. I am Jihad Yassin, a medical student at the University of Jordan. Today we'll be exploring the applications of synthetic biology from like constructing synthetic gene circuits to creating custom organisms. This field is like certainly revolutionizing applications in medicine, industry, and even energy. So yeah, as you can see here, in synthetic biology, we basically design and even construct biomolecular components, networks, or even whole pathways like biosynthetic pathways. So we have applications in, for example, biosensing, creating like sensors and transducers from biological modules and components, therapeutics such as cancer therapy, and even biomanufacturing and pharmaceuticals. Synthetic biology has like its roots in the analogy between like electrical circuits and biological regulation. One of the earliest breakthroughs was the genetic switch or the genetic toggle switch, and we also had the repressolator. These like early innovations proved that we could engineer cells to store like memory and keep time, just like digital circuits or digital electronics. And these examples have showed us that biology can be modular, biology can be functional, biological systems they can be programmable if we have sufficient understanding, and they could be predictable in terms of their behaviors. So we can now talk about the two foundational elements of like both electronics and biology, which are switches and oscillators. Starting with the switches, in electronics, one of the simplest memory devices is called the RS latch. As you can see here, the reset set latch. It's basically a bistable system. It's built like from logic gates. Here, these are called the NOR gates. And it can hold one of the two stable states, flipping back and forth based on the inputs it receives. Once it stays in the state until a new input tell it to change. So this is called bistability, and this is a form of memory. So once it is in one state, it keeps on that state, even if we remove the original stimulus until we induce another stimulus, it will switch to the other state. And interestingly, we see a very similar concept in biology, like biological systems use bistable switches for like critical decisive processes, such as when a stem cell decides what type of specialized cell it's going to become. A well-known example is the natural, like a PRM switch in the lambda phage virus. You know, like viruses with their replication cycles, we have the lytic cycle, we have the lysogenic cycle. This system mainly relies like on mutual repression. As you can see here, we have like two proteins that are produced and they repress each other from their promoters. Depending on which gene is like dominant, the virus will either destroy the host cell, which is known as the lytic pathway, or integrate into the genome and lies dormant, which is known as the lysogenic pathway. Building on this idea, um, researchers have created a synthetic version of the genetic toggle switch using the same modular components, and they called it like the genetic toggle switch. Um, it basically uses two promoters, one from the lambda phage, which activates a gene called the LAC-I, and we have another one called the PTRC2, as you can see here, which drives the temperature sensitive version of another gene, which is known as the CLTS. Each of these represses the other, just like the natural switch. So as you can see here, we have mutual repression. When we add a chemical, like for example IPTG, or we increase the temperature briefly, we can flip the switch. And most importantly, just like in like, for example, electronics, it remembers its state even after the signal goes away. So if we relieve the IPTG signal, it will retain its current state until we induce another signal or another stimulus. It will change to the other state within this bistable system. So that's basically a synthetic biological memory. Now let's move into the oscillators. Oscillators are very essential for keeping time in both electronics and biology and circadian rhythms. In electronics, this might be something like the capacitor inductor oscillator, or the LC circuit, which is a loop of an inductor and a capacitor that produces like rhythmic signals. In living organisms, we see natural oscillators everywhere, like from sleep-wake cycles to plant photosynthesis. One amazing biological example is the circadian clock in cyanobacteria. It is a very well-known example in systems and synthetic biology. It's basically powered by feedback loops and 3 genes. We have the Chi-A, we have the Chi-B, we have the Chi-C, which creates daily rhythms in the cell's behavior. And so, inspired by this, famous synthetic biology researchers Illowitz and Liebler built a synthetic oscillator called the repressilator. Instead of the natural clock genes, the Chi genes, they use a loop of 3 repressor genes within a transformable plasmid, each one turning off the next in a cycle-like manner. And this simple design produces like a repeative wave of gene expression, which can be observed in terms of the amounts of the products of these genes. So as you can see here, we have the repressilator genetic circuit devised by Illowitz, Michael Illowitz. We have here the 3 proteins, 3 repressor proteins, they repress each other. We have here the plasmid that we transformed into the bacteria, and the expression of each gene is linked to the expression of a certain fluorescent marker. For example, here we have green fluorescent protein, red fluorescent protein, blue fluorescent protein, and they keep inhibiting each other. They have a certain cycle with a certain time or a certain, for example, half-life, and they keep oscillating. One of the exciting applications is biosensing applications. Cells naturally detect environmental cues, for example, toxins, temperature changes, and they respond. So we can utilize these capabilities and engineer these capabilities using, for example, DNA, RNA, or even proteins to build biosensors that are highly specific to their targets. Whether it's, for example, E. coli detecting pollution, or RNA switches, riboregulators responding to metabolites, the possibilities are like various and vast. So you can see here we have synthetic biosensors integrating environment responsive promoters. Usually when we talk about sensors in, for example, electrical engineering, we have a responsive element, and we have the transducer, which detects and responds, and which actuates the signal into an effect or a response. We have different types of biosensing applications. We have transcriptional biosensing at the level of transcribing genes, translational at the level of translating the genes, for example, riboregulators or RNA-based switches. They detect certain intracellular metabolites, and then they regulate the translation process. And we also have post-translational biosensors, like engineered protein receptors. So yeah, we can see here the examples of the synthetic biosensors, which are foundational tools in synthetic biology. At their core, as I said before, we have the sensitive element that recognizes and binds a specific molecule, and we have the transducer element, which converts this molecular recognition into a measurable signal. We can look at the two major types of biosensors. We have the transcriptional and we have the translational biosensors. Starting with the transcriptional biosensors, they are basically built by linking the environment responsive element or the environment responsive promoters to engineered gene circuits. One of the good examples is the transcriptional AND gate, which only activates the production of a certain product when two different or two distinct environmental signals are present at the same time. So this is the function of the AND gate. So we have two ones or two ons, and it produces an output. These, for example, environmental cues or signals could be, for example, salicylate and arabinose. Upon the presence of both salicylate and arabinose, the output is produced. So how does it work? For example, in this example, one signal, for example, arabinose, it triggers the promoter known as PBAD to begin the transcription of the T7 RNA polymerase gene. This gene, however, it includes stop codons known as the AMBER stop codons, which block the translation process later. So now we have the salicylate, which activates the sub-D, which activates, the PSAL promoter, which turns on the expression of a special tRNA molecule known as the sub-D tRNA molecule that suppresses those AMBER stop codons. So upon the expression of both of these genes, the mRNA molecule will be translated successfully. So we need both signals, salicylate and arabinose. So you can see that it functions like an AND gate. So only when both signals are dictated does the full RNA polymerase get translated and become functional, allowing it to trigger a downstream output gene, which could be technically anything you need. In this case, it could be, for example, linked to a GFP signal, and we can observe the fluorescence. And that's how we get precise control. It's not enough for just one signal to be present, both must be present. This design, as we said, synthetic biology proves the modularity of biology. So this design is also modular, meaning that we can swap out, we can change the inputs, we can change the outputs, but the main idea, the main contextless module is the same. So we could redesign the entire circuit. Now into the translational biosensors. These work a little bit more different. Instead of relying on transcription and promoters, they use RNA structure to control whether a protein is made and whether a protein is translated. A very great example is the anti-switch biosensor, which includes two domains. In one RNA molecule, we have an aptamer. We have the aptamer domain within the RNA molecule, which binds to small signal molecules, like for example here we have thiophylline, and we have the antisense domain of this RNA switch or this anti-switch sensor. The antisense domain can block the translation by binding on a target mRNA. In the absence of thiophylline, the antisense domain of the RNA switch or the riboregulator is hidden. It's basically locked inside a stem loop structure, as you can see here. So it's hidden and locked by stem loop structure. Upon binding of thiophylline, a conformational change is induced in the structure. The RNA changes its shape. The RNA regulator changes its shape, freeing this antisense domain from the stem loop, as you can see here. So it's able to bind to the target mRNA molecule, and it blocks the translation of the downstream gene, acting as an off switch. So it works at the level of the translation controlled by RNA regulators. This kind of biosensor provides a very powerful and programmable way to control gene expression with high specificity and modularity, as we said before. So we can change thiophylline into another thing. We can change the riboswitch itself, the structure of the riboswitch itself, to respond to other stimulus and to control another set of mRNAs. Okay, so these were like examples of transcriptional and translational biosensors, and we saw how flexible they are, how programmable they are. Okay. Uh, so as you can see here, we have the third type of the synthetic biosensor, which is the post-translational biosensor. And we have also the hybrid architecture, which involves different types of biosensor modules linked together. So starting with like the post-translational biosensor, as you can see here, we talked about this, the sensitive element, the transducer element of a biosensor, which is the first design principle. For the post-translation biosensor, in this type, the sensitive element is typically a membrane-bound protein receptor. So we design a specific receptor, and nowadays we have a lot of, for example, artificial intelligence tools that enable the design of certain receptors with certain responsive domains. When this receptor detects an external signal, like a hormone or a molecule in the environment, it triggers a signaling cascade inside a specific signaling cascade inside the cell, for example, the MAP kinase pathway, and etc. So yeah, a great example is based on the yeast MAPK pathway, which naturally controls response to like mating signals in yeast. Researchers have engineered a synthetic protein scaffold. As you can see here, we have a synthetic protein scaffold that gathers all of the signaling proteins within a pathway into a single platform. And why do you think is this important? It's because in biochemistry we have a thing known as compartmentalization or localization of the components. For example, when talking about the components of the electron transport chain, they are compartmentalized. This localization of the components within a unit allows the fine-tuning of the response of the system. Like if you want a faster reaction or maybe a delayed or a highly sensitive one, you just like modify the structure of the scaffold, or you adjust which proteins it can take or recruit. Yeah, so this approach also gives a modular and a programmable way to control the signal transmission inside the cells, almost like rewiring a circuit board using protein parts. So yeah, we have here the alpha factor, the mating signals, and we have the synthetic scaffold, and so on. And we can adjust this synthetic scaffold. Now let's talk about the hybrid biosensors. Hybrid biosensors, from their name, they are hybrid, so they combine multiple types of like biological modules, transcriptional, translational, post-translational, to achieve even more sophisticated and complicated programmable behaviors. One fascinating example is the synthetic edge detection circuit, which was devised by researchers in 2009 in bacteria, in a lawn of bacteria. Researchers technically created a light sensor by fusing two proteins. We have a photoreceptor from cyanobacteria, the chimeric CPH8 sensor, and we have a kinase from Escherichia coli that is linked to this chimeric light sensor. This chimeric sensor, called the CPH8, turns on in the absence of light. So in dark, it turns on and it activates a certain promoter. As you can see here, we have the promoter OMP-C. So cells in the dark, they start producing a signaling molecule called AHL that is downstream of this promoter, and a transcriptional repressor known as CL. As you can see here, CL is a transcriptional repressor. Meanwhile, cells in the light do not produce these. As we said, this receptor is activated in the dark. So this cells in the night will not produce the transcription repressor and will not produce the AHL molecule. But if they are next to a dark cell that releases, for example, AHL, they can respond. So if we have certain cells or certain bacterial cells that are exposed to the light, they don't produce the molecule, but this AHL molecule is diffusible to the cell, from the cell, is diffusible from the cells that are exposed to the dark into the cells that are exposed to the light. And why is this intelligent, actually? Because the system is wired so that only the cells on the boundary, on the edge of the image, those in the light but near the dark, will activate a certain pigment production, as you can see here. The result, bacteria can literally trace the edges of a projected images, just like the image processing techniques that are used in artificial intelligence and electronics, forming patterns like biological photography. So yeah, this example shows how creative we can be with biology and synthetic biology, and we can introduce novel functionality. Biological systems are not designed to for edge detection, so we have introduced a novel function for biological systems. So yeah, this is the paper. You can read this interesting paper. It was published in Cell in 2009. As you can see here, we have the circuit, and we can even write the pseudocode for this circuit. We have the HL molecule, the diffusible molecule. We have the CPH8 dark sensor, and so on. And we have the production of the black pigments, or the black pigment by the cells at the edge. They have compared also the outputs of the in vivo bacterial system, bacterial lawn system, and the outputs of the mathematical model. As you can see here, we have the photography element, we have inverter element, edge detector element, or edge detector biological circuit, and so on. And just like computers, as you saw before, just like computers use logic gates, the AND gates, the OR gates, the NAND gates, cells can be engineered to do the same. We can design genetic circuits, processing multiple inputs to give precise outputs. This allows us to make cellular decisions based on complicated or complex environments. Yeah, so synthetic biology, it has, other than the biosensing applications, it has some applications in therapeutics, and it's basically reshaping medicine. We can now, for example, engineer some systems or cells to identify drug targets, for example. Synthetic circuits can help identify new drug targets. It could be used in drug discovery to screen for new antibiotics and compounds, or even for the delivery of therapeutic delivery. For example, bacteriophages can be modified to find antibiotic resistant bacteria, evade certain resistant mechanisms within antibiotic resistant bacteria, and engineered viruses can selectively, for example, kill cancer cells, for example, oncolytic viruses. So yeah, we are basically entering a new time, or a new era of programmable medicine, basically, and we have, we'll see some prominent applications in cancer immunotherapy related to synthetic biology. So, okay, so let's now have a look at how synthetic biology is transforming the full spectrum of therapeutics, from discovering new drugs to delivering them more precisely, and finally to produce them at scale. We started with disease mechanism and drug target identification. For example, understanding the mechanism of agammaglobulinemia, which is the lack of gamma immunoglobulins, by introducing certain BCR or B cell receptor constructs into a synthetic system, and introducing rare mutations into this BCR construct, we can observe for the phenotypes such as agammaglobulinemia and the lack of assembly of the immunoglobulins. So yeah, this could give insights into the mechanism of agammaglobulinemia and mutations in BCR. Another thing is drug target identification. One example was from a study which involves the use of riboswitches and riboregulators to control the expression of a certain toxic molecule in bacteria, which inhibits DNA gyrase. and from this experiment, they could deduce, for example, the mechanism of antibiotics. They discovered, for example, that most bactericidal antibiotics work by mechanisms of oxidative damage and oxidative stress and so on. In terms of, for example, drug discovery, as you can see here, we see a synthetic mammalian gene circuit. It is a synthetic gene circuit in a mammalian cell that is used to find treatments for a bacteria, tuberculosis. The challenge with the antibiotic ethionamide, which is used as a treatment for tuberculosis, ethionamide has to be activated by an enzyme known as ETHA or ETHA to work against the M tuberculosis or the MTB. However, the bacteria has certain evasion mechanisms. The bacteria often escapes treatment because ETHA is naturally repressed by a protein known as ETHR. So to solve this problem, researchers have engineered this synthetic system. They have fused the ETHR with the transcription activator protein VP16, and this complex activates the expression of the SEAP protein, or the secreted alkaline phosphatase, which is a reporter gene, through the PMEN promoter. If we now introduce a candidate antibiotic molecules into the system, we can screen for good ETHR inhibitors to relieve the compounds that relieve ETHA repression and restore ethionamide's effectiveness can be discovered. And what is better with this system is that we are using this synthetic gene circuit within a mammalian cell. Because this whole system runs in mammalian cells, the screen also helps identify drug candidates that are permeable and non-toxic to the mammalian cells themselves. And these are two critical traits for success of an antibiotic. Another example is the control of therapies, therapeutic control and delivery. We can see here the transcription system, a synthetic gene switch designed for precise and reversible control over gene expression. It is useful in gene therapies and drug delivery. The switch works at two basic levels. We have the transcriptional level, which is blocked by the LAC-I repressor. So as you can see here, we have the LAC-I. The LAC-I represses through a reporter, or an operator site in the gene of interest that is used in the gene therapy. Here we have an off switch. If any messenger RNA leaks through, it could be also silenced at the translation level by a special RNAi, or an RNA interference mechanism, as you can see here, by targeting the 3' UTR region. The smart part here is the introduction of the IPTG molecule. The IPTG molecule, we add the small molecule like IPTG, and both blocks are lifted. When we add the IPTG, we relieve the LAC-I gene block, and we relieve the RNA interference translational block. So yeah, we introduce an on switch under the introduction of IPTG. So we can have, and this gives us like a tight, tunable and reversible control over the gene that is used for gene therapy. Another final example, and a very important example, is drug production and access. Even if we discover, for example, the active compounds in drugs, we have another big challenge, making the drugs affordable, making the drugs accessible to ensure equity in healthcare, especially for prevalent diseases. Antibiotics, for example, they are relatively cheap. They are produced within microbes and fungi naturally, but some critical drugs such as artemisinin, there is an antimalarial drug, the ACT-based therapy, or even Taxol for cancer, it comes from plants or rare sources that don't scale well. Synthetic biology is solving this problem or changing that. We take artemisinin as an example. Artemisinin is part of a large family of natural products known as isoprenoids. Scientists can bypass the natural inefficient DXP pathway, the deoxyxylulose phosphate pathway in Escherichia coli, by building a complete mevalonate pathway from yeast directly into Escherichia coli. So as you can see here, we have modular parts we are playing with, and we are using these modular parts in different organisms other than the native organism. So we optimize each segment. We have the top operon acetyl-CoA into the mevalonate. We have the bottom operon mevalonate into the farnesyl pyrophosphate, and we link these top operons, bottom operons, like we test them in piecewise manner. We link them, and we optimize each segment of the pathway, known as the top and the bottom operons, and we then link them to a codon optimized enzyme known as ADX. You know, like, what do I mean by codon optimized here? Sometimes different codons, they differ by their last base, and they could encode for the same amino acid. So sometimes we could optimize the codon for highest yield and highest translational efficiency. We have an codon optimized ADS enzyme that is important for the production of artemisinin precursor from the product of these two segments. Once the entire system is debugged and refined in E. coli, they can move back the system into yeast for industrial scale production. So now we have a cost-effective and high-yield platform for producing artemisinin precursors. One of the recent applications in cancer immunotherapy includes CAR T cells. CAR T cells are immunotherapies mostly used in hematological malignancies, and they were proven to be very efficient in the treatment. So we basically collect the blood of the patient, we extract, isolate the T cells, we reprogram the T cells to have chimeric antigen receptors, we multiply these cells and infuse the cell into the patients. These CAR T cells can recognize cancer cells, recognize certain new antigens, and kill the cancer cells. Several synthetic genetic switches or synthetic switches have been programmed in CAR T cells to control them. For example, here, if antigen A is present within the cancer cell and induced CAR, so we target antigen B and we kill, for example, the cancer cell. We have AND gates, we have OR gates. We could have, for example, certain genetic switches that can deactivate these CAR T cells to reduce the side effects of CAR T cells, such as the cytokine release syndrome, the neurological side effects such as ICANS, and so on. Now for the biofuel and industrial applications of synthetic biology. In energy and manufacturing, synthetic biology can allow for the production of biofuels and pharmaceuticals from microbes. Yeast and bacteria can be redesigned to produce biofuels such as ethanol, butanol, or even anti-malarial drugs such as artemisinin. We can also use engineered cells to create sustainable biomaterials and bioplastics. So we have microbial production of biofuels, pharmaceuticals, as well as biomaterials. To efficiently produce biological products, we need to fine-tune the gene expression. We have different strategies. We need to fine-tune the gene expression of the product within the biosynthetic pathways. This can be achieved by re-engineering the promoter to control the gene expression, scaffolding proteins for optimizing and metabolic flux or the flux of metabolites, or even directed evolution. The utmost goal is optimizing each step in the pathway to increase the yield and efficiency while reducing the waste products. So in terms of controlling metabolic flux, evolutionary strategies and rational design strategies could be utilized. When it comes to producing complex molecules like artemisinin, for example, pathway optimization is key. Two major strategies have emerged. As I said, we have the evolutionary combinatorial strategy and we have the rational model-guided strategy. Starting with the evolutionary one, in producing artemisinin precursors, scientists originally avoided using the native DXP pathway within E. coli, the native E. coli isoprenoid pathway, because it was tightly regulated and very intrinsic to the bacteria and difficult to reprogram. Instead of abandoning the pathway or leaving this pathway entirely, Some researchers used a clever technique known as multiplex automated genome engineering, or MAGE, which basically introduces small targeted mutations across the genome using synthetic DNA nucleotide. So we have oligonucleotide-based allelic replacement that takes place in rapid cycles to generate vast genetic diversity across the DXP pathway genes that encode the biosynthetic enzymes. Through this process, the researchers evolved versions of the DXP pathway that had significantly higher translational efficiency. So it's like an evolutionary strategy. We have variation, and we have fitness, and we have the survival for the fittest. Yeah, and we have lycopene. Lycopene is a compound which is brightly colored, and it could be measured. So we can optimize for the production of lycopene. The power of this technique is that it allows in vivo directed evolution. It is fast, automated optimization of this complex pathway. Now, looking at the rational design model-guided strategy, at the opposite end of the strategy spectrum is the model-driven design. It's not based on trial and error. Rather, it's based on quantitative models and parameter tuning. It's based on mechanistic approaches. Imagine like you have a promoter, or you have an RBS, a ribosome binding site you want to optimize. You want to test the circuit. You collect within a synthetic system, you collect the input-output data, and you fit these input and output data into a model, and you find the best parameter fit. The model, like, it estimates the best fit parameters, such as the reaction rates, the strength of expression, or the binding affinities to the promoters or the binding sites. Yeah, then using these insights, using these models, we can forward engineer new components and new entire pathways that meet the specific performance goals, and that are based on the parameter sets of these models that are based on modular components. This approach is being used now in stochastic modeling of gene expression to help predict how engineered promoters will behave in living cells, and they are used also in thermodynamic models, so we can forecast or predict the initiation rates of translation of synthetic ribosome binding sequences or RBS sequences. We can predict how much protein will be made from a given mRNA using thermodynamic parameters. So it's like a blueprint-driven mechanistic model. It's not just a random collection of variants and or combinatorial. Now, looking at the hybrid approach for controlling the metabolic flux, and from its name you can guess it's a hybrid strategy that blends both the rational design and the combinatorial optimization design. Here we use like synthetic protein scaffolds to control and fine-tune the metabolic pathways. The approach was pioneered by some researchers, and the goal was to spatially organize and localize the enzymes of the biosynthetic pathway within a unit, similar to how you may organize the tools on a bench for making the job easier. So we have a kind of compartmentalization, localization. They have used certain protein-protein interaction domains like the GBD, the SH3, the PGZ that are derived from certain metazoan signaling systems. These domains are like molecular docking ports. They are like tethered to a certain protein scaffold, and they can bind to certain protein tags or matching peptide tags, and these tags are fused to certain biosynthetic enzymes. In this example, we have three enzymes: the ATO-B enzyme from E. coli, we have the HMGS, which is the HMG-CoA synthase, and the HMGR, which is the HMG-CoA reductase. These are derived from yeast, and these are derived from bacteria, and we can put them in a single synthetic biology system. These three enzymes together form a key part of the mevalonate or the MEV pathway, which converts acetyl-CoA into isoprenoid or artemisinin precursors. Here comes the combinatorial and the combinatorial part of the process, or the evolution part of the process, which is varying the number of repeats within the interaction domains, the number and arrangement of these interaction domains on the scaffold, and the researchers could then tune the relative concentrations or the stoichiometry of the enzymes. instead of globally overexpressing all the enzymes, which is metabolically not cost effective, metabolically expensive for the cell, we achieve a high product yield. We optimize the proximity of the enzymes, we optimize the balance of the enzyme, and we ensure high yield without overburdening the host cell with enzymes. So it's scalable and efficient biosynthesis. Regarding the future challenges and opportunities in synthetic biology, as we scale up synthetic biology, challenges include like expanding the library of our genetic components and genetic parts. We have to improve our computational modeling. We have to develop reliable high throughput measurement tools such as RNA sequencing and the global gene expression measurement tools that could improve the accuracy of the computational models that rely on experimental data to be fitted. We have also to push the boundaries forward into more complex organisms like mammals for medical applications are more relevant pragmatic approaches in synthetic biology. And the takeaway from here is that organisms, biology can sense, biology can compute, biology can act. Computers are not just restricted to digital computers on a silicon chip. Synthetic biology is rapidly changing from theory into practice. Yeah, and hopefully it will address like global challenges. However, we still need our creativity and commitment to this field.